Good afternoon, everyone. Thanks for coming out today. It's a real pleasure to see everybody in person. It's my pleasure today to introduce Dr. Carrie Coleman as today's seminar speaker. Dr. Coleman is an assistant professor in the Department of Microbiology here at UAB. I'll just give a little of the highlights of her very impressive PhD. She got her PhD at LSU Health Science Center in Shreveport. She worked in the lab of Dr. Scott Tibbetts, and that's where she worked on a murine gamma herpes virus, the MHV68. When she finished her graduate training, she stayed with the Tibbetts group and moved with them to the University of Florida Medical College. She continued to work on this murine herpes virus, and did her work in B-cell biology and in some mouse models. And then she wanted to gain some additional training in this area, so she joined the lab of Rosemary Rochford at the SUNY Upstate Medical Center, where she then started her training and gaining her expertise in the Epstein-Barr virus. She did some really groundbreaking work in this postdoctoral fellowship. She established that the Epstein-Barr virus types differ in their cellular tropism. She was able to define the Epstein-Barr virus type 2, unlike the type 1, a unique tropism for T cells, affecting both T and T cell lymphocytes. This was a real change in kind of the thinking of the role of T cells in this virus. We then moved again with the larger Rochford group to Denver. I became a faculty member at the University of Colorado at their medical campus. And there she worked with their mouse core to develop the first Epstein-Barr virus type 2 mouse model, so allowing for in vivo studies for the first time. And then in 2019, in August of 2019, right before the pandemic, she came here and joined the microbiology faculty here at UAB. So she's been very busy setting up her lab. She is leading the journal club in virology for the doctoral students. She's very active in the medical student education program, teaching in virology. And she's also heavily involved in helping develop the humanized mouse model here at UAB. I'm really fortunate to have her here today to share her work. And the title of the talk is Epstein-Barr virus type 2 infection T cells and alternative strategies for viral disease.
Okay, can everybody hear that okay? No weird feedback? Okay. Well, I wanted to start by thanking the organizers for giving me the opportunity to come and visit to this group today. And so just out of curiosity, I found working with this virus, how many people were actually aware that there were two types of EBV? Not a lot. So I've found this out through just interacting with a lot of scientists outside the EBV field, that this is not broad knowledge. There are actually two types of EBV. So today, with my presentation, I would really like people to kind of go away with the understanding that EBV type 2 is actually a relevant human pathogen. And it really should be paid more attention to because it's kind of really largely ignored by the EBV field. And in addition to that, hopefully come away with appreciation for the model, the in vitro and the mice models that we've developed to really study EBV infection, specifically focusing in on the role of T cells in that infection. Okay, so I'll start with just, got it working before. There we go. Okay, so just a little bit of background with EBV. I'm sure you're all aware of this virus, but it is one of the human gamma herpes viruses. It is one of the larger viruses. We're not quite as big as pox viruses, but it's on the larger side with double-stranded DNA viruses. And one of the key aspects is how successful this virus is. It infects up to 95% of the world's adult population. And one of the reasons for that is basically its ability to really establish a lifelong latent infection in its host. And with that, most of us carry around this virus, thankfully, with no severe health consequences. What happens, however, though, in individuals that their immune system can't properly keep this virus under control long-term, is they can develop a number of different lymphomas, and it is classified as a class 1 carcinogen by the World Health Organization. So these lymphomas include those of B-cell origin. We need B-cell, well, I think that showed up anyhow. B-cell origin, there's one epithelial cell origin, nasopharyngeal carcinoma, and then a significant number of T-cell lymphomas and lymphoproof diseases. Now, the majority of these are going to occur in the context, like I mentioned earlier, of a secondary immunosuppression state where individuals with transplants are taking drugs to prevent rejection of the transplanted organ, individuals with HIV that have a suppressed immune system, individuals in Equatorial Africa where malaria is endemic. So people that have a suppressed immune system are most often going to be the ones affected by this particular, by these lymphomas. So it's really a development of these lymphomas subsequent to the virus infecting and establishing a latent infection. So the predominant model in the field that I will start off by noting is based on studies in the US and Europe, healthy individuals and populations where EBV type 1 is the prevalent virus. Okay, so with this model, the overall model for how this virus establishes latency is it infects naive B cells specifically in the oral pharynx. And upon infection of those naive B cells, it expresses a number of different latency genes. So there are actually 9 latency proteins that are expressed in the naive B cell. And these proteins drive the activation of the B cell and drive it to become a germinal center B cell. Once the germinal center is generated by these infected B cells, genes are restricted to latency 2, which is EBNA1, LMP1, and LMP2. So these proteins specifically either maintain the viral episome in those proliferating cells, or the LMP proteins are responsible for making sure that the germinal center B cells survive the germinal center reaction to complete differentiation to become memory B cells. And once the memory B cells, that's where the virus latently resides long-term. And either gene expression is completely shut down, so the virus is completely hidden from the immune system, or under normal homeostatic proliferation of memory B cells, it will express that EBNA1 protein only. And again, that maintains the viral episome in the cell so it's not lost. So this is our overall very strongly held model for EBV establishment of latency with EBV type 1 anyways. But as I mentioned at the beginning, there are actually 2 EBV types, with type 2 really not being studied very well and not having a lot of attention. But the big difference is phenotypically within type 1 and type 2 EBV are within the latency genes. So if you look at the graph here, this is showing the % difference in the genome, and it's showing the location of latency genes at the top here. But the top, the blue line, or green actually, green line and red line, those are comparing a type 1 virus to a type 2 virus. And the blue line in the middle is just comparing 2 type 1 viruses. So there is a significant difference in the genome, and those differences specifically lie within those latency genes. So as you would suspect, with that big of a difference in the genotypes, there's going to be phenotypic differences in these viruses also. So one of the biggest and first noted was the ability of these 2 viruses to actually immortalize B cells in culture. It's really well established that EBV type 1 can readily immortalize B cells infected in culture, or even if you take an individual that their B cells are infected from the PBMCs and culture them, it can also immortalize those B cells. Okay, and it does this pretty efficiently with about 30 to 50% of infected cultures generating an immortalized B cell line, known as the lymphoblastoid cell line from those cultures. Whereas EBV type 2 really doesn't do this very efficiently at all. It's only at the upper end about 2% of those that you will get a transformation event and be able to generate a immortalized cell line with EBV type 2. So this is important because the initial infection and immortalization, so the activation and proliferation of those B cells in culture that results in that immortalization, are thought to really reflect the early stages of EBV independently driving that B cell activation differentiation to become memory in vitro. So the fact that EBV2 can't really do this very well would suggest that EBVs EBV type 2 is impaired in its ability to independently establish that latency in the B cell population. Okay, but despite that, so we have this virus that seems like, based on its phenotypic ability to immortalize B cells, really can't establish latency very well in the human host. But if you look at different populations, specifically in Equatorial Africa and in the HIV positive population worldwide, both EBV types happen to be prevalent in these populations. So you have a significant, so the blue is showing individuals positive for EBV type 1 only. The green is showing EBV type 2 only, and the red is showing co-infection with both EBV types. So we have this contradiction of a virus that doesn't seem to go very well at immortalizing, so suggesting it can't really establish latency independently like type 1, but it is actually prevalent in populations. So how is this occurring, and how is EBV type 2 behaving differently? Okay, and another really important thing to point out, and I think one of the reasons I really wanted to focus in on studies with EBV type 2 is when you look at these populations. Okay, so Equatorial Africa population where malaria is endemic, it has an extremely high incidence of Burkitt's lymphoma. So this population is extremely vulnerable to these EBV lymphomas. And then the HIV positive population is also extremely vulnerable. So these two populations that are more prone to developing these type of malignancies, are the ones where type 2 is prevalent. And if you look at the two populations, if you look at the population with Burkitt's lymphoma and those with non-Hodgkin's lymphoma in the HIV group, and you compare the types associated with those, it's really equal between type 1 and type 2. So it seems in the human population where both are prevalent, they're equally pathogenic as far as their ability to cause these malignancies. Okay, so collectively, we have these viruses that are, behaving differently, but in the population seem to have generally the same outcome and are both very pathogenic viruses. So why is EBV2 able to establish infection and cause that lymphoma to equal amounts as EBV type 1 if it can't really independently establish latency in the B cell compartment like type 1 does? Well, one of the things that we observed is there is a difference in the cell types that these viruses infect. Whereas both EBV type 1 and type 2 are really, really well established infecting B cells and epithelial cells, we have found that EBV type 2 also has the ability to infect T cells. Okay, so I'm going to just give a little bit of background of those initial studies to put everything in context. But this original observation came from Eric Wolford. He was an MD PhD student in Dr. Rochford's lab. And around the time he was getting ready to finish his PhD and go back to med school, he was doing some observations comparing EBV type 1 and EBV type 2 and how much proliferation they were inducing in the B cell population. Because he was also interested in what the difference were in these two EBV types. But what he was doing is he was including T cells in these experiments to look at proliferation, as at this time it was just kind of accepted that EBV doesn't naturally infect T cells, so those shouldn't be proliferating. And obviously for type 1, he was correct, but what he observed in his these experiments was the type 2 infected T cells were actually proliferating to some extent. So as I said, this is about the time he was finishing up in the lab, and this is about the time I was entering Dr. Rochford's lab. So I continued on this observation and did the initial studies looking and further characterizing whether EBV2 was actually infecting the T cells and what effect they had upon infection. And we did find that EBV2, but not EBV1, readily infect primary T cells in culture. And this infection was indeed latent. We got expression of all of the latency genes like you observe upon infection of B cells in culture. The infection of the T cells induced their activation and proliferation. We also got from those T cells expression of a lot of chemokines that aren't the chemokines you would typically see produced in response to EBV. So not a normal immune response to EBV, so something a little bit different going on here. And we also importantly saw that, and we'll get to this a little bit later, that there's preferential infection of CD8 T cells versus CD4. The virus could infect both, but it definitely infected the CD8s at a much higher frequency than the CD4s. And the image here is just showing staining for CD3 and LMP1, which is one of the latency proteins that gets expressed on the surface of the cells. and showing that we see LMP1 positive T cells to confirm that they're directly affecting the T cells. Okay, so based on all of this, my immediate question was, okay, this is great because this provides the very first model, because I gave you that whole list of T cell lymphomas the EBV is associated with in the beginning. But up to this point, the EBV field focuses on EBV type 1. They've taken EBV type 1. They've tried to infect T cells in culture. They haven't been able to do it. So there was just no model available to study EBV infected T cells or how the etiology of any of those lymphomas that are EBV associated, right? So this provides a great in vitro model to start understanding the etiology of those EBV associated T cell lymphomas and lymphoproliferative diseases. But I also really wanted to know, is this just something happening in a dish, or is this actually happening in the human population? So one of the reasons I joined Dr. Reicher's lab is she also has a lab in Kisumu, Kenya, which is an area with a really high prevalence of children that develop Burkitt's lymphoma. So I made a trip to Kenya to take on this particular study. And here I showed you the studies that have been done in Equatorial Africa, where we know that EBV type 2 is prevalent in that population, but that's really focused on the adults in that population. And the study that Rosemary had, Dr. Rochford had going, was basically following children from birth through 2 years of age to look at infection, when they get infected and how that affects their immune system as far as like what relates to Burkitt's lymphoma development. So we had the availability of these samples that we could analyze, but first I wanted to determine whether the children were also co-infected with both EBV types. So with that, I took 35 of our PBMC samples that were collected, and specifically here we tested at 12 months of age and looked for EBV type. We did typing for EBV type 1, type 2, and then co-infection, and we found a significant percent were either type 1 only positive, but then we had a number that were type 2 only positive or co-infected with both. So that would mean that these samples provide a way to address whether these T cells are infected. And at this point, none of these children have had any development of Burkitt's lymphoma yet. So these are healthy individuals, healthy children in the Kenyan population that we can look into. Okay, so with this, we took PBMCs from 12 of our children, and we chose 6 that at 12 months of age were only, their PBMCs were only testing positive for EBV type 1, and 6 that their PBMCs were only testing positive for EBV type 2. And then from those PBMCs, what I did is I isolated using magnetic columns the T cells. So I purified the T cell fraction. And because we're collecting from children, we can't get a whole lot of blood, and we need to do multiple experiments with these PBMCs. So I couldn't really have enough PBMCs to isolate both T cells and B cells specifically. So we isolated the T cells, and then we also collected the non-T cell fraction, which would contain the B cells and should really be the only cell type present in that fraction that would be infected by EBV. So as you would expect in the non-T cell fraction that contains those B cells, we were able to detect EBV in all 12 of the fractions. And in the first 6, like we saw in our full PBMCs, it was EBV type 1. And in our child 7 through 12, our second 6 set, those were also solely positive for EBV type 2. So that's really what we expected. What we were really surprised and excited to find is when we looked at those T cell fractions from those same kids, all 6 of the kids that were positive for EBV type 2 and their PBMCs were also, their T cells were positive for type 2. So extremely excited about that, proof that type 2 is actually affecting T cells in healthy people. One result that I was a little puzzled about and didn't know initially kind of what to make is one of our children, child number 1, his PBMCs were only positive for EBV, total PBMCs positive for EBV type 1. His T cells showed up also positive for EBV type 2. So I didn't quite know what was going on here, but then we looked back at the data and the other time points of the blood collections and the typing, and what we found. Oh, okay. Well, I'll just tell you what we found. Basically, I don't know what happened there. So at 14, we tested. This shows at 14 weeks, 6 months, and 12 months for this particular child. And what we found is at 14 weeks, this child was actually positive for EBV type 2, and then later switched over to where we can only detect type 1 in the PBMCs, which we see that often in that Kenyan population of switching from one type to another detected in circulation, and we don't know exactly why that's happening. But this child was indeed positive for EBV type 2 at an earlier age in total PBMCs. So we think that's why it's potentially showing up in the T cells, but that also suggests that the infection T cells might be, like it is in the B cells, a long-term infection. It might be persisting in that T cell population. So we really wanted to, or at least I wanted to address that next. And one of the ways I thought to do this is when we were taking the samples from the children, we also took samples from the mothers of these children. And since these are adults, the virus would have had kind of a longer time to actually establish a true latency. So we looked at the mom. Oh, I jumped ahead a little bit. Sorry about that. We did look at later time points in our T cells because of this, suggesting it was a later time point. We took the very same kids, their PBMCs that were collected at 24 months, and we also isolated those T cells and non-T cell fractions. All the non-T cell fractions remained positive, but you can see that we had some of them that, again, switched over from being able to detect that type 2 to only being able to detect type 1 in the non-T cell fraction. So we did have some changes there, but we did still see 3 of our children that were originally had the T cells positive that remained positive. So the virus is able to persist in that T cell population for up to 2 years. So that kind of suggests it could either be kind of a long-term transient infection where over time the infected T cells are eventually cleared by the immune system, or it could be they really are long-term infected, but the frequency of infection is dropping below the level that our PCR can actually detect. So to kind of address this, we then looked at the mom. And with the moms, we, again, could detect EBV in all of the non-T cell fractions. So again, that B cell containing fraction. And the majority of them, we detected EBV type 1 in circulation. In that population, 2 of them, we could detect type 2. But we didn't detect, unfortunately, any in the T cell fraction. So that still really left both options open. It could be more of a transient infection that's cleared over the first few years after infection, or it could be more of a long-term that just is below our level of detection. But we had some other samples available that I was interested in looking into from the moms. We had saliva and we had breast milk. And the reason we collect those is because it's really well established that's how EBV is transmitted, is through saliva. But Ibrahim Dowd, one of the other students, it was actually one of the students in Kenya, had done a study looking at breast milk and these moms, because when they are exposed through pregnancy to malaria, it can really reactivate the virus. So in this case, we saw a lot of viral shedding in the breast milk, and the children in this population are infected at a really early age. A lot of them are EBV seropositive by 6 months of age. So they're getting infected much earlier than the kids in the US or in Europe or other developed countries would. So we were looking at these, and what we found is the EBV type that we could find in both the saliva and the breast milk really well correlated with what we were detecting the PBMCs of our children at that 12 months of age. So that kind of confirms that this is probably the most likely method of transmission, but it also brings up this, we're seeing these moms that were really only predominantly detecting EBV type 1 in circulation, but they're definitely infected with EBV type 2 because it's present in their saliva and breast milk. So that kind of, in addition to our 2 options of either kind of a transient infection or long-term infection, maybe we're just completely missing it. Maybe we're looking in the wrong place, because with this, it suggests that maybe type 2 is residing outside of circulation. It is more of a tissue resident virus, unlike type 1 that is known to reside in circulation. So with this, I think it's possible that we might be underestimating the prevalence of type 2 in all populations if we're just not looking in the correct place. If we're only ever looking at PBMCs, but EBV type 2 is predominantly residing outside of circulation, then we might just be missing it. And a study that actually kind of supports that idea, it was one shortly after EBV type 2 was discovered. Dr. John Sixby at St. Jude tested a number of different workers at St. Jude. But what he did differently is he didn't look at PBMC. He actually did throat washings because he was really interested a lot in viral reactivation. So he took throat washings of a number of different patients. It was like 150 something, and 34 of them showed up positive for EBV. And when he typed those, he found 41% were actually EBV2 positive. And then 9% were infected with both type 1 and type 2. So that's a much higher prevalence that he's finding in this particular adult population in the US than they found when they looked at PBMCs from a lot of areas in the US and in Europe. So that kind of supports the idea that maybe we're just not looking in the right place for this particular virus. So what if it is that it does reside out of circulation and it's in populations that are going to have a lot of inflammation and potential EBV reactivation, like areas where malaria is endemic, or in the HIV positive population that you're potentially going to have a lot of inflammatory events, that's why we're able to actually detect the virus in those particular groups of people. So to kind of carry that a little bit further, one population that we know has a really high reactivation rate of EBV is MS patients. So I had found this paper that they had done in Spain where they were looking at the prevalence of the two EBV types in the healthy population versus the MS population. And what they found there is, although they do see a higher prevalence of EBV type 2 in that population in general, overall, they had 90% of MS patients in that population that were co-infected with both EBV types. So kind of along these lines, I wanted to see if this was also true for the US population. So US is estimated to have about 5% of the population positive for EBV type 2. So with that, I worked with Enrique Alvarez, who is the MD that runs the MS Center in Colorado, who has a really high population of MS patients. And so I got 20 PBMC samples from what they had stored for healthy age-matched controls, and then 20 PBMC samples from MS patients enrolled in the clinic there. And these are specifically MS patients that had not undergone any B-cell depletion therapies as of this point in time when the PBMCs were collected. And what we found with this is 60% of that particular population was co-infected with both EBV types. So again, maybe inflammatory events are what make these people more susceptible to infection with both EBV types, or maybe it's what's allowing us to actually detect the EBV types in circulation. And again, we're just underestimating the prevalence. So this is really one of my major points of interest with EBV type 2, is what the real actual prevalence is in the population. And so one of the things that I wanted to do is really try to get to addressing where the virus is actually, where type 2 actually resides in a host. So to do that, one of the things I really need to do is to develop a humanized mouse model, because we're going to be limited in the types of experiments, especially tissue-specific experiments we can do in humans, right? So there have been a number of different EBV type 1 humanized mouse models already developed, but not one for EBV type 2. So with that, I worked with Roberta Polinda and Julie Lang, who ran the humanized mouse core in Denver. And the reason I specifically wanted to work for them is they had a humanized mouse model that had really high efficient engraftment of the human immune cells in a mouse model without the need for additional tissue transplants in these mice. So all we do is basically take our mice and inject them with human stem cells, and they get around a 50 to 80% engraftment of those human B cells and T cells. Okay, so, and if I'm not going to go, this is one slide. If you're interested in the mouse model for any reason, let me know. I'm happy to talk about that further. But with this, basically, we established this model demonstrating that, like we see in vitro and in our human population, EBV type 1 or type 2 infects both B cells and T cells, whereas when we infect with type 1, we only get infection of the B cell population. But also, like we see in the human population, we got development of diffuse large B-cell lymphoma in both mice. So mice infected with EBV type 1 or EBV type 2, around 75% of these mice with either strain of virus develop these lymphomas. They develop at the same rate, so they develop about 5 to 8 weeks post-infection. And we did further histological analysis of those tumors and found out it was a plasmablastic form of diffuse large B-cell lymphoma, and with very striking similarities. So we didn't see a lot of differences in the tumors developed in the EBV1 versus EBV2. One of the differences we did see that was really interesting to me, though, is which mice actually developed the tumors. So if you compare mice with tumors and without tumors, and what the percentage of T cells were, human T cells specifically in those mice, we found that, kind of as you would expect what we know about EBV type 1, the fewer human T cells they had, the more likely they were to develop a tumor. Whereas the more human T cells they had, the less likely they were to develop a tumor. That just makes sense because that's what we know about EBV. But we saw the exact opposite of that with EBV type 2. The fewer human T cells, no tumors, more human T cells, higher incidence of tumors. So that really suggests that infection of those T cells is contributing to the development of those lymphomas and may be a critical part of EBV type 2 actually establishing latency. Okay, so basically with all of those studies, putting our in vitro studies, our human studies, and the humanized mouse model all together, we've developed a working model in which basically we suggest that EBV type 1 or type 2, sorry, is infecting T cells, and with that infection, it can infect both CD8s and CD4s, but just to a different extent. And that's a latent infection. That latent infection drives the activation and proliferation of those T cells and the expression of a number of different chemokines. We think with the infection and potentially the expression of those chemokines, that's contributing to the establishment of latency in the B cell compartment. And we think also based on our humanized mouse model that the T cells are important for development of these lymphomas. Now that may be just solely the T cells are required to establish latency in the B cell compartment because that's obviously a prerequisite to the development of these lymphomas. Or by production of these chemokines, it could be contributing to the inflammatory environment within that tumor. So those are things we really want to use our mouse model to do depletions at different times of infection to determine how these T cells are really contributing. Are they contributing to establishment of latency? Are they contributing directly to tumor development or potentially both? And then obviously one of the things I really want to address with this mouse model is what are the long-term reservoirs and where are they residing? Because even if a T cell is not a long-term reservoir, are those B cells that are long-term reservoirs residing more in tissues than in circulation? So for this, we want to harvest the tissues from the infected mice and use the comet, the Luminor comet that's in the Shelby flow core that can basically do multicolored histological analysis to see if we can identify the very specific subsets that are EBV positive through different stages of infection in our humanized mice. So these studies are in planning, and mostly that's because right now, unfortunately, with these mice, they're severely immunodeficient. They're very prone to infections, and we have a C. bovis infection in our colony that we are putting a lot of effort towards clearing up. My lab manager, thankfully, has a very strong background in bacteriology, so she's been a big help with that. So we're in the process of trying to clean our colony, and we're getting there, but we can't really do experiments with the mice right now. So in the meantime, what we're doing is we're focusing more on our in vitro model and kind of going back to what's going on with the infection of T cells and those T cell lymphomas and lymphoproliferative diseases. Because we want to also bring those potentially back into the humanized mouse model also once we have it working. So for these lymphomas and lymphoproliferative diseases that are EBV associated, they're highly aggressive. Sorry about that. They have an overall really poor prognosis. There's only about 20 to 30% survival for 5 years. And many of them, the standard treatment is from my understanding, talking to Bradley Haverkos, who is an oncologist that specializes in these, R-CHOP is basically the standard treatment. And a lot of them are refractory to that treatment. So prognosis right now for this group of patients is really not good. What we want to do is see if we can utilize our models to hopefully develop therapies that are more specific and have a better outcome. And specifically what we want to do is focus on the EBV latency protein, LMP1, and how its expression is regulated in the T cells. The reason for that is LMP1 is the primary oncoprotein, or LMP1 is the EBV's primary oncoprotein. You can take that protein, express it in murine fibroblasts or T cells, and it'll form cancer, it'll form malignancies in those mice, independent of any other viral protein. So it's very oncogenic. It functions like a constitutively active TNF-alpha receptor. In B cells, it's known to kind of act as a constitutively active CD40, but T cells don't express CD40, so we don't know exactly how it's signaling in T cells, but it is inducing a pretty strong response because we think that's driving a lot of that activation that's occurring in our T cells. It's known to regulate, at least in B cells, a lot of different signaling pathways. So we're doing actually right now, the process of doing RNA-seq analysis and pathway analysis to see if we can specifically define which pathways are driven by LMP1 specifically in the T cells. Okay, so what we know about LMP1. What we know about LMP1 is predominantly from the studies of the B cells. We know in B cells upon primary infection, the expression is regulated by another EBV latency protein, EBNA2. So you get expression of EBNA2 really early within the first day of infection. You can detect the protein, but because LMP1 is driven by that first protein, you get delayed detection of the protein of LMP1. So you don't get that until day 4 to 6 in B cells. But that's in latency 3 when all the latency proteins are expressed. So a lot of lymphomas, so the T cell lymphomas, liver diseases, Hodgkin's lymphoma, nasopharyngeal carcinoma, those lymphomas all express the latency 2 gene profile. So EBNA1, LMP1, LMP2. So you don't have any expression of EBNA2 in those lymphomas. And the thing is, we don't really know how expression of LMP1 is regulated in these scenarios because we don't have any really good models for it. We have a whole bunch of Burkitt's lymphoma cell lines, and that's latency 1, so you only get EBNA1 expressed there. We have a whole bunch of the lymphoblastoid cell lines, and those express latency 3, so all the latency proteins. We don't have any cell lines that will persist in culture that are latency 2. You can get some of the Hodgkin's lymphoma cell lines that are EBV positive to grow out in culture, but they revert to latency 3 very quickly. So with that, it's a challenge to figure out what's really regulating LMP1 in the absence of EBNA2. So big question is, how is it going to be regulated in the T cells? Okay. Wait, stay there for a minute. Okay, so what we did notice that unlike in the B cells, where you don't get LMP1 protein until day 4 or 6, we get LMP1 detected day 1 post-infection. So that would suggest that unlike what's happening in the B cells, EBNA2 is not really regulating expression of LMP1 in T cells, even when it's present. So that means maybe T cells are a good model to, at least in this cell type, understand how LMP1 expression is regulated independent of EBNA2. Okay. What we do know is from studies that there are 2 STAT3 binding sites to the promoter of LMP1 in addition to a STAT5 binding site. In addition to that, so work with these T cell lymphomas that are EBV associated is really challenging. One, we don't have any, until these studies, we didn't really have any models. And working with the actual tissues themselves, the tumors themselves, has been very challenging because they're really highly necrotic. So you can't get a whole lot of information out of those. But some of the studies that have been done have really established that although this is a very kind of diverse group of lymphomas and lymphoproliferative diseases, one thing that seems to hold really common is a activation of the JAK-STAT pathway, constitutive activation of that pathway. So that makes this interesting, knowing that LMP1 may be actually driven by that STAT3 or potentially STAT5 signaling. So we really wanted to look into this, but again, coming back to the difference in the preference for CD4s and CD8s, we can't, so we're really going to focus in on the CD8s because studies with CD4 are going to be extremely challenging because we have a frequency of infection of 1 in 60 of CD4s. So getting enough cells that are infected to actually do those studies is going to be extremely challenging. And we hope we'll get there one day, but right now we're going to focus on the CD8s because those are infected at a frequency of 1 in 3. So we have a much higher frequency of infection. We have a lot more cells that are infected. And the other thing that I think makes that an advantage for us is with this list of lymphomas and liver diseases, the first 4 here are predominantly of CD8 origin. The last 2 are predominantly of CD4 origin, but we have 1 or 2 CD4 EBV positive cell lines, but we don't have any CD8 positive cell lines. So this kind of provides a unique opportunity to look specifically in the CD8 T cell population. Okay, so what we wanted to do for this, because we do get expression of that latency 3 in T cells, even though we don't think EBNA2 is playing a role, we do get the expression of all of them. So we really wanted to look at this specifically as being EBNA2 independent. And we had a great opportunity to do that because we have this EBV2 positive cell line, JOY, it's a Burkitt's lymphoma cell line, And that's how we generate a lot of our virus stocks, is we induce reactivation of the virus from those Burkitt cell lines. So we have the availability of this type 2 virus, and this is what we did a lot of our initial studies on, was the Jejoi strain of virus. But through culturing of the Jejoi strain, there was the generation of a natural deleted mutant virus. So through just culturing of this, there was a deletion of the EBNA2 gene, and that generated the P3HR1 cell line. So P3HR1 is a derivative of Jejoi, it just lacks expression of EBNA2. So we really wanted to utilize that particular virus to do our studies. So we compared frequency of infection with the wild type 2 and the EBNA2 deleted type 2. And we find that with that, we get a really similar frequency of infection, and we get also similar activation and proliferation of those CD8 T cells. So this seems like a great way, like virus, to utilize to look at LMP1 expression completely independent of EBNA2. But before we went too far down the rabbit hole, we really wanted to confirm that there really was no EBNA2 being expressed. So with that, we took our CD8 T cells infected with both the wild type EBV2 with the deleted EBNA2 mutant, and we see that we only get EBNA2 transcripts or protein in the wild type, and with the deleting, there really is no EBNA2 transcript protein detected. So that confirms it really is P3HR1 is really EBNA2 deficient. And when we compare that with LMP1 transcripts or protein infecting with these same viruses, we see very similar levels of transcript and protein, really confirming that LMP1 expression in the context of T cells is EBNA2 dependent. Okay, so now moving on to, is it CD3 or STAT3 or STAT5 that are potentially driving expression of LMP1, or potentially both? So to address this, we just basically took our T cells infected and added STAT3 inhibitors or STAT5 inhibitor at the same time we infected, and then followed them in culture. We continued to add the STAT3 inhibitor every 3 days through our cultures. And what we found is with the addition of 2 different STAT3 inhibitors, we lost expression of LMP1. However, with the addition of a STAT5 inhibitor, there was really not much difference in LMP1 transcript levels. So this would really suggest that STAT3 is the primary transcription factor driving LMP1 expression in the CD8 T cells. Okay, so with that, we really wanted to look, when is STAT3 activated in these T cells, in the CD8 T cells specifically. So we took, this is specifically the EBNA2 deleted virus that we did these experiments with. So we infected, and we started looking as early as 15 minutes post-infection and initially out to an hour. And we saw phosphorylated STAT3 as early as that 15-minute time point. So the phosphorylation of STAT3 and the activation of STAT3 is happening very, very quickly in these infected T cells. And it doesn't have to be active, intact virus for that to happen, because we UV inactivate our virus and we get the same thing. We still get phosphostat3 by 15 minutes, maintained out to about an hour. So it's initiating activation of STAT3 very quickly. And this obviously doesn't seem to need to be an active infection, which would indicate that it's probably viral binding that's inducing that very early STAT activation. But we also want to look, how long is that STAT3 activation sustained? And so we looked from day 1 out to day 12, and we got a really strong signal for phosphostat3 through that entire time point. So it looks like not only is STAT3 very rapidly activated, it's sustained activation in these T cells. Okay, so then the question becomes, what's inducing the STAT3 and what's sustaining the STAT3? So studies done by Nick Smith, who was a student in Rosemary's lab, his focus was identifying what the glycoprotein was used by the virus and what the receptor was on the T cell that allowed type 2 to actually infect. So to do these, he basically used a whole bunch of neutralizing antibodies provided by Lindsay Hupfletcher, who had done a lot of these studies with B cells, that neutralized the glycoproteins that are known to be required for B cell or epithelial cell infection. And when he added the neutralizing antibody for GP350 to the virus before infection, that's what blocked infection of the T cells. So we know from the studies with B cells that GP350 binds to CD21. So he took a jerk-kit T cell line and did a number of different CD21 knockouts, and that also demonstrated that CD21 is required. So it's GP350 and CD21 that are required for EBV type 2 to infect these CD8 T cells. So there might be other co-receptors that are not yet identified. We know at least these, this viral glycoprotein and this T cell receptor are required for infection. Okay, and the reason I get to that is because he also compared the level of CD21 on the T cells that are CD4 positive, CD8 positive, and you get a much, much higher concentration of CD21 on the CD8s. So that's probably why we have that different preferential infection, a much higher frequency of CD8s infected compared to CD4s. So when we repeated our experiment and did infection of CD4 with the virus and looked at phosphostat-3, we get a really, really low level of phosphostat-3 detected with the CD4s. So that kind of further supports the idea that it's probably viral binding, GP350 binding to CD21, that's driving that initial STAT3 activation. Okay, so then moving on to what do we think is sustaining STAT3 activation. So we know that our T cells, specifically the CD8, doesn't really happen with the CD4s. The CD8 produce a lot of chemokines. Specifically, RATs, MIP1 alpha, MIP1 beta, MCP1, and IL-8 are produced at a really high level in the supername of these CD8 T cells that are infected. So basically, we know from other studies that these very same chemokines can be driven by LMP1. So expression of those can be driven by LMP1 in B cells or epithelial cells. So that suggests that probably in our T cells, LMP1 is playing a big role in regulating the expression, because remember, these are chemokines that are not normally expressed in response to an EBV infection. So to kind of get at this, we took the same STAT3 inhibitors. We took our T cell infected with the, this is going to be the EBNA2 deleted virus. We infected, and then we infected with the presence of the STAT3 inhibitor. And when the STAT3 inhibitor is present, we get a significant reduction in the production of those chemokines by our CD8 T cells. So that, again, not a direct, we are working on that experiment to directly determine that LMP1 is driving expression of these, but this is really supportive of the idea that LMP1 is driving expression of these chemokines in our T cells. Okay, also, just as a note to kind of tie into the model I'm about to present, the receptors for all these chemokines are expressed on T cells both prior to infection, and the expression is maintained after infection. We do get some drop in expression in a few of those, but for the most part, they're pretty consistent expression levels in infected and uninfected T cells. Okay, so that kind of brings us to the model that we're really focusing a lot on right now, and we're doing experiments to test. We think that the virus GP350 binding to CD21 on the CD8 T cells is driving the initial activation of STAT3. How it's doing that is a big question, because CD21 is one of those proteins that really has to pair with other proteins on the cell surface. It has a very short cytoplasmic tail and doesn't really signal well on its own. It's known to couple with the B cell receptor on B cells, but we don't know what it couples with on T cells. It's really kind of a black box of what CD21 does on T cells. So I want to work with a lot of the structure people in our department to figure out what potentially CD21 is complexing with on the T cells, and what downstream signaling events can be generated from the protein complex. Okay, so that's a big question we have here, but we think that that's driving the initial STAT3 activation, and that STAT3 drives the initial expression of LMP1 in those CD8 T cells. Once LMP1 is expressed on the cell surface, it acts as that tissue-active TNF-alpha receptor signaling, and that potentially we think is driving the expression of our chemokines. So in the B cells, epithelial cells, the expression of those is driven by the NF-kappa-B signaling pathway. We don't know if that's true in our T cells. We got to test that. So that's some of the work we're doing with the gene expression analysis.
I'll ask you a question here.
Yeah.
I think you mentioned that LMP1 functions as CD40.
In B cells, but we don't know, this T cells don't express CD40. So, and CD40 is a TNF receptor, so we just know it probably functions as some kind of TNF receptor on T cells. So we don't know if the signaling is going to be the same.
But the CD40 is like this single transmembrane receptor. This is 7.
Yeah, but
very different.
Yeah, but it's really well established that LMP1 acts as a tissue-active CD40 for B cells, and LMP2 acts as a tissue-active B cell receptor. That's how it drives that activation and proliferation initially. But again, we don't know how it's behaving in T cells. So because these are new models. But then we think that the chemokines that are expressed act in an autocrine fashion to slowly back on those receptors and maintain that STAT3 activation, because it's been shown in T cells, at least 3 of these, RANTs, MIP1 alpha and MIP1 beta can activate STAT3 through that signal. So this is our model that we're testing. We're doing a lot of blocking antibodies to see if we can block these interactions. My lab manager, Melissa Oliver, has generated a whole bunch of lentiviral vectors to use to test this model. So we're in the process of doing all these studies to really rigorously test this particular model, with the goal of really identifying, hopefully, some targets we can then go into our humanized mice and test to see if we can block LMP1 expression in vivo. Because targeting STAT3 is a possibility, but that's going to come with a lot of extra problems. So if we can block something like CCR5, a receptor that's involved in that sustained signaling, then maybe that's a safer option for developing a treatment. So that's the overall goal for these studies, is to get into our humanized mice to try to have some safer targets. They're never completely safe, but safer than just targeting STAT3. Okay. So with that, just thanking everybody. Melissa is kind of my right-hand person in the lab currently. She is really doing a lot to try to get our humanized mouse colony clean. She's great with all the genetic stuff and generating our lentiviral vectors. So she does a ton of things in the lab. We've also had a lot of help from Jan Novak and Stacy Hall. They do a lot of glycoprotein production. So they've helped us generate soluble GP350 to do some of our studies. And then obviously there's a lot of people from Rosemary's lab that were involved in the initial studies here. Nick was really involved in the receptor research. And then Roberta and Julie, or the humanized mouse core lab, were huge in developing that model. And then Eric was the one that was the student that made that very first initial observation. And obviously I have to thank all the donors, both from Kenya and from our PBMC donors here. We also collect a lot of tonsils and things like that. So all the donors, and then the funding sources for all these studies. So with that, I'm going to take any questions.
Fascinating. A number of patients with BTLD improve just by decreasing the anti-rejection medication. Mm-hmm. How can we explain that on this model?
Well, I mean, if that's specifically type 1, which it may be a lot, and again, we don't know the prevalence of type 2 in the US, and so if that's predominantly going to be a type 1 driven, that is known to very much need that CD8 T cell response to be properly controlled. So it would make sense that alleviation of that immunosuppressive would then be able to control the virus.
But so in PTLD, EBV1 is responsible, not EBV2?
So I am not sure. I don't know. I don't think I've seen studies that looked at EBV type specifically in post-transplant lymphoproliferative disease. So whether their type 2 is involved in that, I don't know.
The result you showed from the MS studies was really interesting.
Yeah, I'm trying to follow up on that with some more.
I'm kind of hoping that you pursue that. I mean, how do you want to take that, or what do you think the key things are?
Well, so I've got a R21 in now for that. So what I would like to do is basically be able to do analysis of a much larger patient cohort to really establish the percentage in that. Also, there's some really interesting studies that have shown that EBNA2 can basically act as a transcription for a lot of genes that have EBV risk or MS risk alleles. I believe there's like 34 of the genes it associates with. But the EBNA2 for type 1 and type 2 is really different. It only is about 50% homology similarities in those two, and they've been shown to drive very different just general gene expression in just B cells. So one of the other things I would really like to do is compare the EBNA2 from type 1 and type 2, and then both of those collectively in the expression of those genes with those MS risk alleles.
Look at the R21.
Thank you.
So is there, I guess, look at the EBV positive T cell. Mm-hmm. Has people determined if it's type 1 or type 2 EBV?
It can be either. And we think so. We think what's happening, it really depends on what population they're coming from. And we think what's happening is potentially in certain scenarios that there's an upregulation of CD21 on those T cells that allows EBV type 2 in some scenarios to infect. Because it seems with comparing CD4 and CD8 that the level of CD21 on the cell surface has a big impact on whether the cells are susceptible to infection or not. So we think that that might be how type 1 is actually, type 1, we think it's more of a accidental infection, whereas with type 2, it seems like it's actually part of the viral life cycle to infect the T cells. But the T cell lymphomas, lymphoid diseases can be associated with either type 1 or type 2.
And in your mouse model, do you ever see T cell lymphomas?
We don't, but that's another thing we're working on. What we want to do is, because the B cell lymphomas develop so quickly, what we want to do is actually infect our mice, let latency be established, but before they start to develop those B cell lymphomas, deplete the B cells and see if we can give it time for those T cell lymphomas to develop in our mice.
Any other questions?
Yeah.
One last question.
Yeah.
I'm curious. So on the individual cell level, do one cell get infected by both types of viruses, or is it just once you have one, it rejects the other?
No, so once you get infected with one, it does downregulate CD21, so it might be harder to get co-infected, but there has been shown intertypic recombination events occurring between the two types, so you can get infection of both types in the same cell. It's possible. How often it occurs, I don't know if it's established.
Well, great. Well, let's thank Dr. Coleman for